Gram stain. Gram stain or gram staining, also called Gram's method, is a method of staining used to distinguish and classify bacterial species into two large groups, gram-positive bacteria and gram-negative bacteria. The name comes from the Danish bacteriologist Hans Christian Gram, who developed the technique. Gram staining differentiates bacteria by the chemical and physical properties of their cell walls. Gram-positive cells have a thick layer of peptidoglycan in the cell wall that retains the primary stain, crystal violet. Gram-negative cells have a thinner peptidoglycan layer that allows the crystal violet to wash out on addition of ethanol. They are stained pink or red by the counterstain, commonly safranine or fuchsin. Lugol's iodine solution is always added after addition of crystal violet to strengthen the bonds of the stain with the cell membrane. Gram staining is almost always the first step in the preliminary identification of a bacterial organism. While gram staining is a valuable diagnostic tool in both clinical and research settings, not all bacteria can be definitively classified by this technique. This gives rise to gram-variable and gram-indeterminate groups. The method is named after its inventor, the Danish scientist Hans Christian Gram, 1853–1938, who developed the technique while working with Karl Friedlander in the morgue of the City Hospital in Berlin in 1884. Gram devised his technique not for the purpose of distinguishing one type of bacterium from another but to make bacteria more visible in stained sections of lung tissue. He published his method in 1884, and included in his short report the observation that the typhus bacillus did not retain the stain. Gram staining is a bacteriological laboratory technique used to differentiate bacterial species into two large groups, gram-positive and gram-negative, based on the physical properties of their cell walls. The gram stain is not an infallible tool for diagnosis, identification, or phylogeny, and it is of extremely limited use in environmental microbiology. It is used mainly to make a preliminary morphologic identification or to establish that there are significant numbers of bacteria in a clinical specimen. It cannot identify bacteria to the species level, and for most medical conditions, it should not be used as the sole method of bacterial identification. In clinical microbiology laboratories, it is used in combination with other traditional and molecular techniques to identify bacteria. Some organisms are gram-variable, meaning they may stain either negative or positive. Some are not stained with either dye used in the gram technique and are not seen. In a modern environmental or molecular microbiology lab, most identification is done using genetic sequences and other molecular techniques, which are far more specific and informative than differential staining. Gram stains are performed on body fluid or biopsy when infection is suspected. Gram stains yield results much more quickly than culturing, and are especially important when infection would make an important difference in the patient's treatment and prognosis. Examples are cerebrospinal fluid for meningitis and synovial fluid for septic arthritis. Staining mechanism: purple-stained gram-positive and pink-stained gram-negative. Gram-positive bacteria have a thick mesh-like cell wall made of peptidoglycan, 50–90% of cell envelope, and as a result are stained purple by crystal violet, whereas gram-negative bacteria have a thinner layer, 10% of cell envelope. So do not retain the purple stain and are counterstained pink by safranine. There are 4 basic steps of the gram stain. Applying a primary stain, crystal violet, to a heat-fixed smear of a bacterial culture. Heat fixation kills some bacteria but is mostly used to affix the bacteria to the slides so that they don't rinse out during the staining procedure. The addition of iodide, which binds to crystal violet and traps it in the cell. Rapid decolorization with ethanol or acetone. Counterstaining with safranine. Carbylfuchsin is sometimes substituted for safranine since it more intensely stains anaerobic bacteria, but it is less commonly used as a counterstain. Crystal violet, CV, dissociates in aqueous solutions into CV and chloride, Cl, ions. These ions penetrate the cell wall of both gram-positive and gram-negative cells. The CV and ion interacts with negatively charged components of bacterial cells and stains the cells purple. Iodide interacts with CV and forms large complexes of crystal violet and iodine, CV, I, within the inner and outer layers of the cell. Iodine is often referred to as a mordant, but is a trapping agent that prevents the removal of the CV, I complex and, therefore, colors the cell. When a decolorizer such as alcohol or acetone is added, it interacts with the lipids of the cell membrane. A gram-negative cell loses its outer lipopolysaccharide membrane, and the inner peptidoglycan layer is left exposed. The CV, I complexes are washed from the gram-negative cell along with the outer membrane. In contrast, a gram-positive cell becomes dehydrated from an ethanol treatment. The large CV, I complexes become trapped within the gram-positive cell due to the multilayered nature of its peptidoglycan. The decolorization step is critical and must be timed correctly. The crystal violet stain is removed from both gram-positive and negative cells if the decolorizing agent is left on too long, a matter of seconds. After decolorization, the gram-positive cell remains purple and the gram-negative cell loses its purple color. Counterstain, which is usually positively charged safranine or basic fuchsin, is applied last to give decolorized gram-negative bacteria a pink or red color. Both gram-positive bacteria and gram-negative bacteria pick up the counterstain. The counterstain, however, is unseen on gram-positive bacteria because of the darker crystal violet stain. Gram-positive bacteria. Gram-positive bacteria generally have a single membrane, monoderm, surrounded by a thick peptidoglycan. Some bacteria have cell walls which are particularly adept at retaining stains. These will appear positive by gram stain even though they are not closely related to other gram-positive bacteria. These are called acid-fast bacteria, and can only be differentiated from other gram-positive bacteria by special staining procedures. Gram-negative bacteria. Gram-negative bacteria generally possess a thin layer of peptidoglycan between two membranes. Most bacterial phyla are gram-negative. Gram-variable and gram-indeterminate bacteria. Some bacteria, after staining with the gram stain, yield a gram-variable pattern, a mix of pink and purple cells are seen. Gram-indeterminate bacteria do not respond predictably to gram staining and, therefore, cannot be determined as either gram-positive or gram-negative. The term gram staining is derived from the surname of Hans Christian Gram. The eponym, gram, is therefore capitalized but not the common noun, stain, as is usual for scientific terms. The initial letters of gram-positive and gram-negative, which are eponymous adjectives, can be either capital G or lowercase g, depending on what style guide, if any, governs the document being written.